All right, so let's get started. Welcome. Good morning, good afternoon, good evening, depending on where in the world you are joining us from, and warm welcome to all of you. On behalf of entire ImmPort team, thank you for being here today for ImmPort End-to-End Workshop. It's a hands-on workshop on data submission, sharing AI-powered analysis, AI-powered access and analysis. My name is Sanchita Bhattacharya. I'm the Scientific Outreach Program Lead for ImmPort. I'll be moderating the session today along with my team members. It's a support from the team: Emma Kenris, Jaya Cannon, Kate Hypes, Sammy Smith, Srinivas Chapuri, Ruth Montero, Matt Strubb, Parisa Pirani, and Julie Tacker. So what we will cover. So ImmPort is for today's agenda, ImmPort is a NIAID-funded, publicly accessible repository for immunology research data. We will follow end-to-end 4 stages: submit the data, discover and access it, connect the AI through MCP server, and then we will finish with guided analysis on an AI-ready data set, and each will be led by the members of our team. 2 quick heads up and some housekeeping items. Before we start, I would like to give some guidelines to the participants. Use a laptop during the hands-on section, keep the laptop ready and follow the instructions from the team members. We will take some short breaks along the way. Have your ImmPort account ready, and it's recommended. Complete if those who got the pre-sessions setup guides, probably you got more instructions with the MCP requisites and so on. During this session, stay muted when you are not speaking, and this session is being recorded, and the recording and the materials will be shared afterwards. Watch the chat we will be posting the links, resources, and instructions throughout. If you have any questions, let's say you have a question, please raise the hand. We will be taking the questions after the presentations and demo, and one of the team members will unmute you. You can also type in the Q&A or chat anytime, and we'll try to address some of them via texting. If you're stuck during hands-on section, send us a chat message. A team member is watching and they will help you directly. Hands-on etiquette, like following along at your own pace is fine. To fall a step behind, that's fine. If you get lost, ask in the chat. So with that, I will start with a quick introduction to ImmPort. Again, those who joined late, I'm Sanchita Bhattacharya. I'm the ImmPort Outreach Program Lead. And today I'll be giving you a quick overview of ImmPort. So ImmPort's mission is to maximize the value of NIAID-funded research, and we accomplish this through 3 activities: archiving the research data, and preserve the primary immunology research data for long-term access, enable the researchers to find and access data sets via the cohort discovery tool, and you will hear more about CDT a little bit later, through the APIs, applied programming interface, and integration with other external search engines. Reuse, we facilitate new scientific discoveries and hypothesis generation by making these FAIR compliant data sets. So we build on the FAIR principles of findable, accessible, interoperable, reusable. And within this ImmPort ecosystem, we have private data space for uploading the data. Shared data is within the ecosystem is through which we disseminate the data to the community. We have a lot of guided analysis, tutorials that's been shared through the resources, and did some limited data analysis as well. And throughout the session, you're going to hear a lot about all of these components. Just repository at glance, we have little over 1,500 share studies as of the release in July. with 194,000 subjects. And this demonstrates the scale of the repository. Um, and we have about like 200,000 subjects contribute to an extensive collection of immunology data sets. And the table on the left, where the breakdowns, like, we support multimodal data, ELISA, ELISpot, flow cytometry, HAI, and many other immunology assay data types. And if you look at the pie chart on the right, we have a lot of different kinds of data sets related to the disease, a lot on immune response, a big 20, like around reaching to 30% now, with immune response, vaccine response, infection response. We have transplant data set, allergy data sets, preterm birth, and many more. As a methods represented across studies, I just mentioned that we support multimodal data, and we have a large amount of flow cytometry data in ImmPort, and then many other assay types. A lot of RNA-seq data, and then spatial transcriptomics are some of those new data types that those are getting deposited in ImmPort. And here is again the breakdowns for different assay types. I have shared the slides, so take a look later. Who submits data? So we have over 35 and more, and these numbers are going up, that NIAID funded programs and beyond, some external data depositors and consortia submitting data to ImmPort. And here is this list, and maybe we have even longer list that I couldn't accommodate on the slide, but you can see that we have variety of data sets coming from consortia and external organizations submitting data to ImmPort. How about the ImmPort data lifecycle? In this workshop is built upon this, like, how this immunology data moves from deposition to discovery. So we have a data submission. You're going to hear about data submission, curation, findability, and accessibility through the web interface as well as through the API, and data reuse. I will not go into detail because Matt is going to cover this, these are the components of the data submission. What do you need to submit the data to ImmPort? As I had mentioned before, we build on FAIR principles, data findable. So we have ImmPort search, cohort discovery tool. You can search for the data sets of interest. Also, we have additional search engines, like data can be accessed through Vivly, NIAID data ecosystem, Google dataset search, and many others. Accessibility, you can get access to ImmPort data sets. What we just require is a registration. So just register to ImmPort, you get a login, and then you're ready to go. You can submit the data, you can access the data right away. And then also through the API, and we're going to cover this a lot today about like different ways the data can be accessed. Fair data principles, interoperability. We are part of the data mesh, and data mesh. This is emerging data mesh framework. We already trying to implement many of the core pillars, which are listed here on the left, and I won't have time to go over all of these, but DOIs, and then metadata, REST API, and so on. And we have started kind of getting into we are interoperating with other repositories and data mesh. Reusable, there is a lot of opportunity for the data sets that's getting disseminated to ImmPort, getting reused. And one of my the group members had been looking at like how we can leverage all the transplant data using multi-agent artificial intelligence framework for reproducibility assessment. And if you are interested for type of data reuse being done, here is this QR code. Please go and take a look at the type of data reuse publications from ImmPort. And if you are interested for this particular data reuse, here is the QR code. We have the preprint on bioRxiv. And that's kind of, in a nutshell, introduction to ImmPort. And so maybe we will take more Q&As, and I'll take a look at the Q&A window if there are any questions. Um, in the interest of time, I'll just hand it over. We are moving to the segment of data submission workflow from Matt and Parisa. And during the Q&A, we can take more questions. With that, I'll stop sharing and hand it over to Matt.
Thanks, Sanchita. I am Matt Strub. I'm the lead data curator for ImmPort. Also want to mention that Prisa Prani is on our curation team as well. So a lot of times if you reach out to the ImmPort help desk, it'll be one of us getting back to you. If you want to follow along with my presentation, you can click on the data submission folder in the workshop materials page, and the presentation is there, as well as a data submission guide that provides some useful information as well. So in this section, I'll talk about working with the ImmPort team, the data submission and sharing workflow, getting started, how to register for a study, how to request a workspace, stuff like that. The study registration wizard, which is a really nice and easy tool that you can utilize to create studies, submission templates, and I'll also show some quick reference guides that are very useful when learning about templates. At the end of this, I will give a brief demo on the website as well. So working with ImmPort, the very first thing you should do is give us a point of contact for all of your data submission communications. I highly recommend requesting a kickoff meeting to walk through the data submission process. That will be either with me or Parisa or both of us. Any questions you have, we can answer, and we can, give you some brief demos to get you started. Just want to remind you, help's always available. Parisa and I answer emails as quickly as we can, and we'll create tickets to assist you in any way possible. And then we also recommend that you share feedback. A lot of the great improvements that we've made over the years are direct results of feedback that we get from users. So we really do listen and we really do incorporate your feedback into the product. You can always email the help desk. This ImmPort_helpdesk at immPort.org is your best friend if you have problems with ImmPort. That goes directly to the curation team and the development team, and we will get back to you. And I also want to highlight that we do have monthly office hours, the first Thursday of every month from 2 to 3 p.m. Eastern. No appointment is needed. You can always drop in there, and we have members of the curation and development and outreach team, and we'll be able to answer your questions. With that said, if you're not able to make monthly office hours, please reach out to the help desk and you can set up a private appointment as well. So briefly, ImmPort hosts two different kinds of data. We call these parse data or study files. Briefly, parse data are structured records that ImmPort extracts from your completed templates and loads into the database. So this will kind of combine your subjects, your clinical data, whether you have lab tests, assessments, etc., your biosamples, and your assay results. It's very accessible by searching and querying through the ImmPort UI and API. And it's really best for cross-study search, filtering, and reanalysis. This does require a bit more work on the data provider's end, filling out templates and whatnot. If you don't want to go through that process, we also do host study files. These are files that you upload with the study. They're kept in their native format and they're not parsed into our database templates. So we can hold raw and result files. For example, if you don't want to parse out your flow cytometry data, we can just host FCS files as study files. We can hold protocols, case report forms, data dictionaries, and any really type of data that's not well captured into a template. And one potential downside to this is that it's not as accessible. You must browse the file manifest and download them. They're not as searchable as well, but they are best for local analysis, source documents in full detail. So with that said, let's go over the submission and sharing workflow. Step one is to register for an ImmPort account. For each of these steps, I do have some subsequent slides I'll show you, and then I will show you in the demo as well. Step two is to request a private workspace. So this keeps your data secure and accessible only to collaborators you designate, and you'll have your own little page with all of your studies. It's very organized, and it helps you keep your studies all in one place. You'll register a study. We recommend using the study registration wizard. You can upload the basic study design, protocols, and basic metadata of your studies. You'll then validate and upload your remaining templates if you are intending to parse out your data. And then lastly, once your data are ready to be shared, you can just reach out to the ImmPort team, again, at that help desk email, or you can email myself or Parisa directly, and we will add your study to the next data release. We currently do monthly data releases, and data is usually due the second Thursday of each month if you want them to go out in that month. So getting started with ImmPort, and again, I'll show you a demo on the actual website after this, but briefly, when you log in to your private data page, you'll see the top orange bar under study. If you click registration, that is how you can start to use the SRW and register a study. Before that, you'll need your own workspace. So if you go up to workspace in the orange bar, you click on My Workspaces, you'll see any workspaces that you're assigned to. If you're not yet assigned to one, you can click Create a Workspace Request. You'll fill out a very brief form and send that to us, and then the curation team will make you a workspace. And this is just what that form looks like. Requesting a workspace, you can either add a user to your workspace or you can request a brand new workspace. We'll ask you some basic questions like grant information and stuff like that. ImmPort is always free to use, but we do track grant information and stuff like that. Managing workspaces, once you do have workspaces, you can manage them, you can assign users to it. You can give special permissions to users. You can see all of your studies. You can update contract grants. For data submission, again, I recommend using the study registration wizard. This is a very cool tool developed by the dev team here, where on the UI you can add all your metadata. You can select your condition and disease, arms, cohorts, etc. And you can actually create a study doing it this way. In the past, you had to fill out a template and it wasn't as user friendly. So I will show you an example of the SRW in a bit here, but I highly recommend it. If you do wish to parse your data, these decision trees are very useful. It'll give you a step-by-step guide on how to upload your templates and in what order. And then we also do have extensive documentation with the data submission templates. And I will show you an example of one of those in a bit here as well. So ImmPort templates. Again, I'll touch on the template upload decision tree. I'll show an assay-specific template ordering. This will be specifically for flow cytometry. And then I'll quickly show you a template quick reference guide. So in this decision tree here, once you've created your study and you want to upload your data using templates, this is the very first decision tree, and it's very easy to follow. First it asks if you have human data or animal data. Sometimes you have both, but if you, for example, only have human data, you'll need to fill out the subject humans.txt template. And then it will ask you, do you have interventions or medications? Do you have adverse events? Do you have assessment data or lab test? If you do, you can absolutely fill out those templates and it will all be parsed to your study. If you don't, you can, completely ignore them. And following that, you will upload any of these templates that you fill out. And once you do that, you can go on to your actual assay templates. So this is a workflow for flow cytometry. And again, we host many assay types. It's one of the unique and powerful things about ImmPort is we take a lot of different types of data. So you can find every assay's decision tree at docs.immPort.org/data submission. We also do have a generic other template that can parse out data that doesn't fit any of the assigned assay templates as well. But for flow cytometry, you'll see in this workflow that we do require 3 templates at least. There's a treatments template, a reagents template, and an experiment samples.flow template. We also do offer a reagent sets template, which is just a way to kind of organize the reagents a bit easier, but that's not required. And you'll see that you'll upload these 3 or 4 templates along with your FCS files. And once that upload's completed, if you want your derived data to be parsed into an ImmPort template, you can do the FCM derived data template and upload that as well, but that is optional. So for each assay type, we do have these workflow diagrams. I highly recommend looking them over. If you try to upload just your experiment samples.flow template, for example, you will receive an error. So it's best to, read up on these assay types and flow diagrams a little bit better. So this is an example of one of the templates that we'll fill out, and I'll go over this a bit more in the demo section. But this is the flow cytometry experiment samples template. You'll see that there's lots of columns here. Some of them are color coded to keep things kind of combined here. For example, your experiment sample ID, your reagent, your treatment, and your FCS result file are all blue here. Those are all related in the relational database. You'll see experiment ID and protocol ID and stuff like that are combined as well. It's not super important to understand that, but I will say that anything that is bolded here usually means that it is required. And if the text is not bolded, it is usually optional. But you can also go on the documentation site and find the page for this template, and it will tell you exactly what is and what is not required. I also want to highlight that if you hover your mouse over these cells, the column headers, you'll get a description of what it is looking for and whether or not it's free text or if you have to pick something from the dropdown. And we do have ontology-driven dropdowns for some columns in which you are required to select one of the terms. Um, if you feel that a term that best describes your data is not found in these dropdowns, you can let the curation team know, and we're actually able to add them for you. So you can download these templates by going to resources, data submission, and submission templates. You can download all of them as a zip file if you want. You can download them individually if you don't want to download all of them, but you can see that the required data column on the far right will tell you which columns are required. And if you go down even further and click on template documentation and templates, each template has its own page and it will give you a description of what each column is looking for and whether or not it's required. Lastly, once you do create a study, whether it's with the SRW or the basic study design template, you're automatically given an SDY accession number. That will never change, and we actually recommend that when you're citing import in a manuscript or a grant, you use that SDY number. That's going to be very easy for data reusers or anybody reading your manuscript or grant to find them, and we recommend using the line, the data supporting this publication is available at ImmPort under study accession SDY, and then your numbers. If you were to create a study right now, we are in the SDY 3000 somewhere. So it'll be a little bit higher than 1, 2, 3, 4. To summarize this before the live demo, you can register for ImmPort and request a private workspace. You can register your study. I recommend using the SRW. You're also very welcome to use the basic study design template. You can complete, validate, and upload your submission templates, and I'll show you how to do that in a moment here. And then you'll share your study in the next ImmPort data release. So now I'll do a live demonstration of some of this stuff. When you go to immPort.org, this is the home page. A lot of the data submission stuff will happen in your private data page. So that is this one here. You can click on search private data. And if you do not have a workspace yet, again, you can click on this top orange bar here, workspace, create a workspace request. You'll fill out this form here. So create a new workspace. It'll ask, are you part of an existing consortium? Is there a grant associated? We ask for a timeline if you have one on, when you're looking to get your data shared. Are you the administrator and so forth? You'll submit that. It goes right to the curation team and we will create a workspace for you. If you ever want us to add users to your workspace, you can fill out this request as well. And once you have a workspace, you can click on My Workspaces and you'll see all of your workspaces here. Most people only have a few. I have a lot because I'm on the curation team, but usually you'll have, a few here. And if you click on it, you can see all of your studies, whether or not they're shared. You'll see what users belong to that workspace and what contracts and grants belong to it as well. Once you have a workspace, if you want to register a study with the study registration wizard, you can click on study on the top orange bar here and click on registration. This will take you to the SRW page. You'll select your workspace. So we have a kind of a curation sandbox workspace here, and you can click new study. And this is what it looks like. So first you'll add basic study information, um, titles, what your sponsoring organization is. It'll ask for descriptions, endpoints, if there's an intervention agent, stuff like that. We'll also ask for a planned public release date. If you don't really have one, you can just put it far into the future. And I would like to note that the planned public release date does not ensure that it's going to go out then. So, if you put that you want it out at the end of August, you'll still be required to let us know when you do want it released. This is really just kind of for our tracking purposes, but it's not an official agreement that we'll release it then. The next tab will ask for categorization. So in this kind of mock study we're setting up here, it's immune response. It'll ask for a condition or disease. Here I picked type 1 diabetes. The next breadcrumb here is arms or cohorts. So in this, mock study, I've created 4 arms here. We have type 1 diabetics who are unvaccinated and those who are vaccinated. And then we have non-disease individuals who are unvaccinated or vaccinated. It'll ask if you have any inclusion or exclusion criteria. So here, I entered an age lower limit and an age upper limit, and then also you must have an A1C greater than 7%. It will ask for personnel. You'll need at least one personnel. This will be the point of contact. You can add as many as you would like. Some people, if there's 10 people on a study, they will add all 10, but we do at least require one. We ask if there's any planned visits or time series. So here, I've just had visit 1 will be an influenza diagnosis, and visit 2 would be a blood draw. If you don't have planned visits or a time series, you can click that box there. We do require at least one protocol. This can either be for an assay. For example, I put the flow cytometry protocol here, or if you have like a case report form for a clinical trial or something like that, you could upload that here as well. We do ask you to link a grant to your research study. If you have publications already available on PubMed, you can add your publication here. And if you have any other relevant links that you would like attached to your study, you can add those here as well. And then, as I mentioned before, the second type of data that we accept are study files. If you want to add some study files directly in the SRW study creation here, we have an option where you can do so here. Once this SRW is filled out, and I think this usually takes anywhere from a half hour to an hour, you can click finish and submit to import. Once you do that, it'll show up here, it'll be completed. And once it is showing as completed, you can click upload. I'm not going to do that here. I will simply, pretend that we're uploading it. It takes a few minutes, but to save time, I will go back to my workspace and I will show you what it looks like once that has been uploaded. So if you go to study search, workspace name, you can look up your workspace. Ours is curation sandbox. And you'll see that the study shows up here. So this is the one we just created. It's assigned STY 3740. And if you click on it, it'll open up the page where you can see all of your metadata. You can go to design and see those arms that we created, the planned visits, the inclusion criteria, etc. And if you click on the study files tab on the far right, you'll see that that's study file that we uploaded in the SRW is showing up here. Briefly, I do want to go over how to parse out experiments. So in this mock study here, we have flow cytometry experiments. And as I mentioned before, those specific templates are required. So I'll pull some of those up and walk you through them. The treatments template is one of the required templates. It's very easy. There's only really 3 required columns. One is a user-defined ID, and this is really just for mapping purposes on our end, but it's also a good way for you to organize your data using any nomenclature that makes sense to you. Here I just did treatment one, but again, it could really be anything. And since I'm not having a treatment in this experiment, I put no treatment. And then for use treatment, I put no. If you do have a treatment, you would fill out user-defined ID. It could be as simple as treatment two, whatever your treatment is, and then you would select yes. And then it would require you to fill out at least one of these blocks here. So it could be a mount value, for example, if it's a small molecule. If there's a specific duration, you could include that. And if there's any temperature adjustments, you could put that there as well. But in this experiment, since there's no treatment, we will leave it at this. The next one that is required is reagents. So in this experiment, we're only going to use the one reagent. It'll be CD4 as a user-defined ID. Again, that could be whatever you want it to be. It asks for a name, the manufacturer, catalog number. The analyte reported is a drop-down, so you can select CD4. If you have an analyte that is not in the drop-down, again, you can reach out to us and we will add it for you. And then it will ask for a clone name and a reporter name. And then lastly, well, not lastly, but two more templates. One is the subject humans template. Again, the subject ID is just for mapping purposes. It can be whatever you want. It'll ask you to assign that individual to one of your arms or cohorts. So again, you can either use the user-defined ID that you used for your arm, or you can use the arm accession number that was assigned to your study. So if you go to design, you'll see that you have your user-defined ID. This is what you selected, or this is what you determined when you were filling out the SRW. Since the study's been uploaded, it was assigned an ARMA session number, so you could use that one if you preferred. Either the ARMA session or the user-defined ID is acceptable. It'll ask for sex. This is a drop-down where you have to select one of these as well. A minimum subject age, age unit. The age event here will be age and enrollment, but there are, different options you can select there. Subject location is required. I kept it brief and selected the country, but if they are within the United States of America, you can get as specific as the state level if you want to. Ethnicity and race. And then on the far right here, it'll ask for exposure process reported. In this mock study, it's individuals that have been exposed to influenza. So I picked occurrence of infectious disease. The exposure material reported is the influenza A virus. If your exposure material is not here, again, let us know. And since we selected something from the drop-down here, we do not have to fill out the exposure material ID because when you select something from the exposure material reported column, it will automatically fill this in. As you can see here, the NCBI taxon ID is there. Disease reported is type 1 diabetes. Again, since we selected it from the dropdown, it already has the DOID here, so we don't have to fill it out in the next column. And then the disease stage reported is unknown. And the last template we'll fill out is the experiment samples template. Again, experimental sample ID, the biosample ID, and the experiment ID can be whatever you want. Those are for mapping purposes. It'll ask what reagents from the reagent template are used in each individual sample. If we go back to the reagents template, you'll see that our user-defined ID here is CD4. So we can use that user-defined ID in this template here, and we'll link them together. Likewise, with the treatments template, our user-defined ID is treatment one. We'll use that to attach it to the experiment samples template. We'll then tell the template for each experimental sample here what FCS file should be associated with it. So we've made some just dummy FCS files here. It'll ask what study ID this is for. So again, once you created your study using the SRW, the study was assigned SDY 3740. It will ask for protocol ID. You can either do your user-defined ID, or again, if you go back to your study's details, and you go down to protocols, you can find your protocol accession number here. So that works in the template. It'll ask for your subject ID. Again, in our subjects template, we use the nomenclature subject 1 through 5 in the subject ID. So we can take that and use it here. If there was a planned visit for this experiment sample, again, you can go back to planned visits in your study page, or you can use your user-defined ID. If you do not have planned visits, there will be a planned visit that shows up here called a dummy planned visit, and you'll actually still be assigned a PV number. Please use that in your template. When your study is publicly released, it won't show up as having a planned visit, but this will satisfy the template. You'll pick what type of sample it is. Here we're doing serum, a study time collected, 3 days. There's a study time event, time of enrollment, hospital admission, etc. Here it's just a time of a blood draw, so I selected other, and then since I selected other, I will specify it in the next column, time of blood draw. If you selected something else here, like time of enrollment, you would not be required to fill out column W. And then you can name your experiment and select the measurement technique. So once you have all of these, the next step is to validate them. And let's actually create some errors here on purpose. So let's remove reagent ID for this, and let's get rid of this FCS result file. And then on the subjects, maybe we'll delete an age for somebody. And in the reagents, let's get rid of a reporter name. So now we know that these templates are incomplete, but say, it's your first time filling out templates and this is what you have. You forgot some things. I'll show you the validator tool. Very handy. I recommend doing it before any uploads. On the orange bar here, you'll go to validate, validate data. This is our validator tool. You'll look up your workspace. Again, ours is Curation Sandbox. Package name could be anything you want it to be. Let's do flow test. You'll select the files. So here I have these 4 templates. They're all saved as text files, which is required now. Although in the future, I think we're going to allow Excel files as well. And then um, let's upload 4 of the 5 biosamples. So we should have 5 FCS files. Here we only have 4. And we'll do a validator. This takes anywhere from, a minute to a few minutes, depending on how large it is. And to save time, okay, so here you'll see that it rejected your validation, which indicates that something is wrong with your templates. You can download the database report, which I've already done, and I will open for you and show you how to kind of navigate these. So these can be intimidating when you first look at them. There's a lot of information here. It's basically showing a step-by-step overview of what the software is doing. But I think the easiest way to find any errors are just to select all, go to conditional formatting, and ask it to highlight any text that contains error. So we'll see that a few are here in the reagents.flow template. There's an error. It says there's a missing required value, and it says that the column is reporter name. That makes sense since we deleted that reporter name. If we go down, there's subject humans, another error, missing conditional required value, and it's saying that there's no minimum subject age. Again, that makes sense. We deleted that on purpose. And then for experimental samples.flow, I see another error. It says there's a missing conditional required value, and it says it's in reagents. That makes sense. We deleted reagents from them. And then lastly, it says that a file does not exist, and it's saying that the flow subject 5 biosample1.fcs file is missing, and that makes sense as well because we did not upload it. So you'll be able to go back to these templates, fix all of the errors. So we'll add back CD4. We'll go back to the human subjects. Oh, there's a missing age, fixed it, and we'll go back to reagents, and the reporter name is missing, and we'll save that. Once you've made the corrections, you can do the validator again. If everything's there, we'll say completed, and that means that everything is good. Once everything has passed validation, you can go to upload, upload data. And there's really two ways to do this. One, if you don't have very large files, you can look up your workspace, make a package name, whatever you want it to be called. You'll select your files and upload. Since I'm doing a flow cytometry experiment, I'm going to use the Aspera method. You'll look up your workspace. And I'll select upload a directory. So essentially, you'll save your templates and your FCS files in the same directory. And you'll select that folder, upload it. I'm not going to do that now since it takes a few minutes, but once your study is uploaded and you go back to your private study page, if you click on the experiments tab here, you'll see that suddenly your flow cytometry data is here. If you click on it, you can go further into it. You'll see that experiment sample accessions have been assigned. There's a row here for each experiment sample. If you click on that, you'll be able to see more information about the biosamples, what reagents are attached to it, what treatments are associated with it, and you'll actually be able to download the result file. So say this is all you want to do with this study, it's ready to be released. You reach out to the help desk or myself or Parisa, and we'll say that it's going to be released at the end of August. This experiment or this kind of mock experiment here is not actually publicly released, but once a study is publicly released, I'll show you on the shared data page. If your study is this, for example, you can look for it. And if you click on it, say this is our experiment, you'll be able to see all of your metadata here, personnel, your design has your arms, your inclusion, exclusion criteria, etc. If you click on study files, if you have any, they'll show up here. And if you want to see your mechanistic flow data, you can click on mechanistic assays and it will show up here. It'll show your protocols, reagents, and treatments. I did want to mention that you can always see your usage metrics for your studies here as well. You can see that this study has had 945 page views. It's been downloaded 55 times via the web and 77 times using the API. And then if you actually want to download your data or data from any study on the shared data site, you can click download study package. It will take you to the download page. Um, all of your metadata is here. If you want to download all of your data, all of your metadata, I recommend downloading this tab file that will have everything. But if you just want to download your FCS files, you can go to results and all of your data is here. And there's also folders for your protocols if you want to download a protocol specifically or any of your study files that you upload. So that is how you would download your data or other data that you're interested in from ImmPort. I think I will pause for questions if there are any. Let's see. Going through the chat here.
Hi, Matt. There is one.
Yes. Has anybody been monitoring the chat? I'm just catching up here.
Yeah, there is one in the Q&A. Um, does the end user need any separate account or license for Aspera to upload by the Aspera mode?
Nope. You just have to download it, and there is a link when you go to the upload or the validation page. We're currently using 4.4.5, so you can just click on the link, and it will take you right to Aspera, and you can download it there. You don't need I don't believe you need a license or anything, and if you do, then it's free. But yeah, it's very simple. And yeah, we'll be updating our upload process in the near future here. And I think we won't be using a spare for much longer, but currently that is the method.
Great. I think there are more questions coming in, and I can read for you. In study file, is there an option to generate a folder, or are there only the predefined folders?
So if you want to organize your study files with folders, the way to do it is to upload it as a zip folder or a zip file, and you can have nested folders within a zip file. But if they're currently unzipped, then we don't have a folder structure for it. But we do have individuals that upload a lot of study files as a single zip file, and the nested folders can be very, very organized.
All right. I think there's one question that I can see in the chat. Can you speak to how OBI fits in with the data model?
Um, yeah, John, can you specify OBI, or does anybody else on the team know?
Yeah, Matt, I might be able to take that one. I know OBI, like the ontology. I'm assuming, John, you're referring to the OBI ontology. And Matt,
Yes.
you can probably speak to how import uses various ontologies, right?
Sure.
Within within the data model.
Yeah, so when we have drop downs that have required ontology, we curate that on our end, and it has to be from a specified ontology source. So there's a few that we use, um, but yeah, some of those columns we do require um, kind of standardized nomenclature. For others, it can be a bit more free text. Yeah, Kate, do you want to add anything to it or?
Yeah, no, Matt, I think you covered it's import uses a variety of ontologies, nomenclatures, and kind of by design to make it use more usable and fit more users' models, I guess. Um, and so I would say too, maybe Emma, if you have it handy, I know we have a link to the ontology and
Yeah,
documentation, I
looks like
believe.
Ruth just added the link.
Okay, perfect. Yeah, and I was like, we can pop that in the chat as well for further exploration. So great.
Yeah,
And then
so
I
this is the fullest ontologies referenced by import. There's several of them, and yeah, we're always looking to follow these, and if there's other sources that are created that are particularly useful, we'll add that to this as well.
Yeah,
yeah,
now
and this is
the link is there now for everyone in the chat if you want to explore the ontologies.
perfect. Thank you, Sanchita. And I see one raised hand. I think in the Q&A, I can unmute there and let's see. Hopefully, there we go. You should be able to speak.
Can you hear me? Can you hear me?
Yes, please go ahead.
Perfect. First of all, thank you very much. Very informative presentation. Um, yeah, I have a question that I also posted in a webinar chat about the possible involvement of AI tools, large language models, or small language models, because it looks like the templates and frames are kind of easy for AI to understand, and therefore, if you create a how-to README that will be readable by AI, and therefore it will become an expert in submission process, then the person can say, okay, what is wrong with my data? What is missing? What is still incompatible with the requirements of import? And um, paste or upload the files to AI instance, then AI, knowing what is required, will read the files and tell, okay, you should fix this, table, or this piece of information is missing, or fix this spreadsheet, and so on and so forth. Right now, it's good, and obviously, the hundreds and thousands, hundreds of thousands of studies that you guys have speak for themselves. But now we have this wonderful AI thing. So did you consider using it to make the submission even simpler? Thank you.
Yeah, absolutely. And I'll let Sammy take this. She can tell you about our AI improvements and goals and plans.
Hi, yeah, excellent question. So short answer is yes, we are working on tools like that. So we did make available within the last year our import documentation chatbot that does have access to all of our documentation, so you can ask questions there. And we do have a lot of information in our documentation about data submission. So as far as any basic questions, our chatbot should be able to handle that in its current iteration. But in the coming year, we plan to extend that to more of a agentic AI architecture that would be able to do things like take a look at your data that you're trying to submit, how you've filled out the template, and give you suggestions on how you might correct any errors that you're running into. There are some security considerations with that, considering that would be data that has not been shared yet. So we want to make sure that users are comfortable with that and opting in only if they would like to use a technology like that. But yes, we do agree with you that that would be really, really useful for our users. So we are exploring adding that capability to our website.
Perfect. Thank you. Yeah. Of course. I share the sentiment that potentially some users might be not comfortable with sharing with AI, but if it is the data that they plan to make public, then I think this lowers the barrier. Anyway, we'll see.
That's a great question. Yeah, sure. So we are working towards that. So in the interest of time, probably let's take a quick 5-minute break. We are running a little bit late, but that's okay. Let's take a quick 5-minute break and then continue. There are some other questions coming. We'll try to type in and to give you the answer. Otherwise, you can also save it for later time. We have a lot of other Q&A segments where we can accommodate the more questions. Thank you so much.